Hello everyone, welcome back to our online lecture series. So in today's lecture we will be discussing about the classification of enzymes. So in last class we have already discussed about the enzyme mechanism with respect to energy. So in today's class we'll see what are the types of enzymes and how they have been classified into different classes. So here you can see the classification of enzymes. Uh, Actually, these enzymes have been classified into 6 major classes. So one is oxidoreductases. So these are the enzymes which will catalyze the oxidation and reduction reactions. And the second category is transferases. So these enzymes will catalyze the transfer of functional groups from one molecule to another molecule. And the third category is hydrolases. So you have heard about the word hydrolysis. So those hydrolysis reactions will be catalyzed by the hydrolases. And the fourth one is the lyases. So these enzymes will catalyze the group elimination reactions to form the double bonds. So and the next fifth one is the isomerases. So isomerases we transfer the transfer the molecule from one isomeric form to another isomeric form. So they will catalyze the isomerization reactions through the bond rearrangements. And the last one is sixth one is the ligases. So these ligases will catalyze the bond formation reactions which couples with the ATP hydrolysis. Why I have mentioned ATP hydrolysis? Because ligases, so these bond formation reactions will require some amount of extra energy. So that energy will be taken from the ATP hydrolysis reactions. So these are the 6 major classes of the enzymes. So now we will see one by one in detail. So here you can see the first class of enzymes, that is oxidoreductases. As I have mentioned in the previous slide, that is, these are the enzymes which will catalyze the oxidation and reduction reactions. So if you see the reaction here, what is happening here, the L-lactate is getting converted into pyruvate. So if you see the structures, the difference in structures in lactate and pyruvate, so during this reaction 2 hydrogens are removed and converting this molecule lactate to pyruvate. So here the enzyme that is being used is the lactate dehydrogenase. So it is taking out the hydrogen. So we know that removal of hydrogen is nothing but the oxidation. So here oxidation reaction is taking place, and that is getting catalyzed, that is being catalyzed by the lactate dehydrogenase. So here NAD is the cofactor which will take part in the reaction. So whatever the hydrogens that are being removed from the lactate, so those will be taken by the NAD place. So NAD is nothing but the nicotinamide adenine dinucleotide. So that is a cofactor. Basically it exists in 2 forms, NAD plus and NADH plus H plus. So NAD plus is the oxidized form and NADH plus H plus is the reduced form. So there are examples for these oxidoreductase enzymes. So those are the oxidases, peroxidases and dehydrogenases. So these are all the enzymes which will come under the oxidoreductase. So the second category is transferases. So these are the enzymes which catalyze the group transfer reactions. If you see the reaction here, there are 2 molecules. One is L-alanine and ketoglutarate. So what group is being transferred here? So the amino group is getting transferred from L-alanine to alpha ketoglutarate. So this is nothing but the transaminase reaction. So the enzyme that is participating in this reaction is the alanine transaminase. So these are all the enzymes which will come under the transferases. So they will transfer one group from one molecule to another molecule. So now we'll see the next one. So next one is the hydrolases. So as I already mentioned that these are the enzymes which catalyzes the hydrolysis reactions. So here you can see pyrophosphate which is having 2 phosphate groups, and these 2 pyrophosphate is getting hydrolyzed with the help of pyrophosphatase enzyme, and it is getting converted into 2 phosphate groups. So initially it is having 2 phosphate groups linking together with oxygen, and during this hydrolysis reaction it will break down into 2 phosphate molecules, and So all these enzymes like esterases, peptidases and glycosidases, these are all will come under the hydrolases. And the next one is, so the next category is lyases. So lyases are the nothing but the enzymes which will catalyze the elimination reaction. If you see here one pyruvate molecule is there, and this pyruvate decarboxylase enzyme which is removing the carboxylate group from the pyruvate, and it is converting this pyruvate into acetaldehyde plus carbon dioxide. So here the pyruvate decarboxylase is eliminating or removing the carboxylate group from the pyruvate molecule. And the next one is isomerases. So isomerases are the kind of enzymes which will catalyze the isomerization reactions. So here you can see alanine, L-alanine and D-alanine. So we know that these two are having the same molecular formula, only the difference is that in L-alanine the amino group is on the left side, and in D-alanine it is on the right side. So already I have discussed about all these things in the regular classes, that L means levorotatory and D means dextrorotatory. So these two are having the same molecular formula, but spatial position is different in these two molecules. So in one L-alanine the amino group is on the left, and in D-alanine it is on the right side. So this is nothing but the racemic mixture of the alanine. So this whatever the enzyme is there, so what it does, alanine racemase, so it will convert the L-alanine to D-alanine. So the next one is ligases. So these are the reactions enzymes which converts the ligation or joining reactions, joining of two substrates. So in this one, the first one is the L-glutamate and the second group is amino group. So ammonium group whatever it is there here, NH4 plus, so this is being added to the L-glutamate molecule. So what is the enzyme here? It is used glutamine synthetase. So this glutamine synthetase is adding the ammonium group to the L-glutamate, and it is converting it into L-glutamine. So as already I mentioned that these are the ligation reactions or joining reactions, or you can say bond formation is taking place in the reaction. So for that bond formation, whatever energy that is required, so that energy will be taken from the ATP. So these are the 6 classes of enzymes, 6 major classes of the enzymes. So with this the enzyme classification part is over. So in the next class we will see the working mechanism of enzymes.